This video is about proteins. Proteins are made up of amino acids. Each amino acid looks like this. Each amino acid has a carboxyl group, an amine group, and an R group. The R group is a variable group. It could have various sorts of groups. It could be a polar group or a non-polar group. There are 20 different types of amino acids. There are 4 levels of proteins. You have the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. I'm going to first talk about the primary structure. Take for example the word near. When you rearrange the word near, you get the word earn. Similarly, if you rearrange different amino acids, you will get different proteins. If the green is alanine, the silver is valine, and the red is proline, you will get one sort of protein. But if you rearrange the structure to mix up all these amino acids, this will give you a totally different protein. Just like when you rearrange words, you'll get new words. After the primary structure, you have the secondary structure. Suppose this is a polypeptide chain and each gem clip is an amino acid. Each gem clip has a CO and an NH group. As they are both polar groups, they start to form hydrogen bonds and they start to coil like this. So the secondary structure becomes a coil structure like this, which we call an alpha helix. A secondary structure also consists of a looser form of an alpha helix called a beta pleated sheet. Here, the hydrogen bonds are along the molecule. The tertiary structure. The tertiary structure is interactions between the R groups. All these variable R groups interact with each other and start to coil. They'll start circling and making bonds and start forming different shapes and proteins. So the 4 main types of bondings for the tertiary structure. You have the hydrogen bonds, the disulfide bridges, ionic bonds, and hydrophobic bonds. Hydrogen bonds. These occur between polar groups. Examples of polar groups would be the NH group, the CO group, and the OH group. As these are polar groups, they form delta positive and delta negative charges. These causes it to form hydrogen bonds. The next sort of bond that occurs between the R groups to form a tertiary structure is the disulfide bridges. These are strong covalent bonds. These usually occur in cysteine molecules. Cysteine is a sort of amino acid. When this arrangement takes place, the hydrogens are removed to form a sulfide bridge. When you add hydrogen to this, you will get back this. The next sort of bonding is ionic bonding. These occur between ionized amine groups and ionized carboxylic groups, as shown in the diagram. The 4th sort of bonding that takes place within R group is called hydrophobic interaction. Hydrophobic means water hating. So this takes place between non-polar groups. As they're water hating, they come together and point inwards, away from the aqueous solution around them. This results in coiling, giving rise to the tertiary structure of a protein. All proteins have a primary and a secondary structure, but only some proteins have tertiary and quaternary structures. Collagen is one such protein that has a primary, secondary as well as a tertiary structure. Here we can see the three polypeptide chains. This is one, the golden one is another, and the pink one is the third. These three polypeptide chains come together and coil to form a triple helix structure. Collagen is a fibrous protein and has a structural function. The last level of protein structure that I'm going to talk about is the quaternary structure. The quaternary structure is the 3D arrangement of two or more polypeptide chains. It could also be the 3D arrangement of a polypeptide chain with a non-protein component. For example, hemoglobin. Here you can see four different chains, polypeptide chains. When these come together with a ion group, which this silver ball here is the ion group, it forms hemoglobin. The ion group is called the prosthetic group, and the four polypeptide chains give it a 3D structure. As these four polypeptide chains have both hydrophobic, water-hating regions as well as hydrophilic, water-loving regions, the hydrophobic regions point inwards. This gives it its tertiary structure. So you can see that hemoglobin has all four levels of proteins. It has the primary structure, the secondary structure, the tertiary, as well as the quaternary structure. Thank you for watching.